Swayam Prabha. Digital India. Educated India.
Welcome to this series of lectures on signal transduction pathways. We just began in the last class where we gave an introduction to signal transduction pathway, and then we discussed how molecules, hydrophobic molecules which can diffuse the plasma membrane, can enter the cell and then activate gene expression through intracellular receptors. We also discussed about molecules which are hydrophilic, therefore cannot cross the plasma membrane. Therefore, they interact with specific cell surface receptors and then transduce signals and then activate gene expression programs. So, what I will do in this class is to discuss it with you about the regulation of gene expression by cyclicAMP. Molecules which interact with cell surface receptors, and this interaction of this ligand with cell surface receptors results in the activation of the GPCRs, that is the G protein coupled receptors, which are the seven serpentine receptors or the 7 membrane receptors, and these G protein activation ultimately results in activation of adenylate cyclase, and adenylate cyclase now produces cyclicAMP. CyclicAMP now activates protein kinase A, and then protein kinase A goes inside the nucleus and activates number of genes. So, what we will discuss in this class is to go in more detail, greater detail, to understand the mechanisms by which cyclicAMP regulates gene expression. So, this is a summary of what we discussed so far in the previous lecture. We discussed introduction to signal transduction. We discussed about how lipophilic molecules transduce signals through intracellular receptors. Then we also discussed briefly how water soluble molecules transduce signals by binding to membrane receptors. Then we discussed about one class of membrane receptors, namely the G protein coupled receptors, and how when the G protein coupled receptors are activated by specific ligands, it results in the activation of adenylate cyclase, leading to a synthesis of cyclicAMP involving the trimeric G proteins. So, let us now discuss and see how does cyclicAMP activate gene expression. Now, cyclicAMP is the first second messenger to have been identified, and it has a very fundamental role in cellular response to a number of extracellular stimuli, and this is the structure of cyclicAMP which I have shown here. You can see it has a 3' 5' cyclicAMP and the phosphodiester bond, so that you have the 3' 5' cyclic monophosphate here. The phosphate is attached to the sugar by 3' 5' phosphodiester bond. Now, cyclicAMP signaling pathway controls a number of cellular processes, and cyclicAMP not only provided the paradigm for second messenger concept, but also provided the paradigm for signaling compartmentalization. As we know, in the cell everything is compartmentalized, and this compartmentalization plays a very important role in the regulation of number of physiological processes. In fact, the credit for discovery of cyclicAMP as a second messenger goes to Harold Wilbur Sutherland Jr., the photograph is shown here, who actually discovered cyclicAMP and then shown that cyclicAMP is a second messenger, for which he was awarded the Nobel Prize in 1971. Now, Sutherland observed that epinephrine would stimulate the liver to convert glycogen to glucose in liver cells, but epinephrine by itself would not convert glycogen to glucose, which means epinephrine is doing this job indirectly, and then he discovered that epinephrine has to trigger the synthesis of the second messenger cyclicAMP for the liver to convert glycogen to glucose. In fact, some of the classical papers published by Donald Sutherland have just shown here in 1958, 2 papers which was published in Journal of Biological Chemistry are listed here, and in fact these were actually reproduced in the centennial year of Journal of Biological Chemistry in the year 2005, where they actually reproduced some of this classical Earl Sutherland's discovery of cyclic adenosine monophosphate under second messenger system. So, the discovery of cyclicAMP as a second messenger is one of the important landmarks in the biological chemistry. Now, cyclicAMP, as I said, is a very important second messenger molecule, and in addition to cyclicAMP there are also many other second messengers which are operating in living systems. Some of them are shown in red here. For example, cyclic GMP is another cyclic nucleotide, as well as inositol triphosphate or IP3, as well as calcium also act as second messengers. In the next class we will discuss the mechanism by which these molecules function as second messengers, and certain hydrophobic or water insoluble molecules like diacylglycerol, phosphatidylinositols, which are membrane associate, they also act as second messengers. So, the G proteins, the trimeric G proteins which are the root cause or which are the anchors which we are discussing right now, and one of the second messengers which are produced by the G protein activation is cyclicAMP. The same G protein signaling can also activate other second messengers like diacylglycerol, phosphatidylinositol, and so on and so forth. This we will discuss in the next class. Now, all these second messengers diffuse from the plasma membrane to the intermembrane space, where they can interact with the membrane associated effector proteins, and many gaseous molecules like nitric oxide and carbon monoxide, which can diffuse to the cytosol and across the cell membranes, also act as second messengers and regulate very important biological classes. So, today we will primarily focus on the mechanism by which cyclicAMP acts as second messenger. In the next class, we will try to discuss the other small molecules like cyclic GMP, inositol triphosphate, diacylglycerol, phosphatidylinositols, and other gaseous molecules, how they function as second messengers and how they activate gene expression programs. Now, what are the hormones which can activate the which when interact with specific membrane receptors result in the synthesis of cyclicAMP? I have listed here. Some of them are epinephrine, norepinephrine, glucagon, ACTH, adrenocorticotropic hormone, vasopressin, luteinizing hormone, thyroid stimulating hormone. There are many others, but I have just given a few examples. Some of them are small molecules and some of them are peptide hormones. So, all these things by interacting with the specific G protein coupled receptors can ultimately lead to the synthesis of cyclicAMP, which in turn goes inside the cell and regulates a number of physiological processes. Now, before we start in-depth discussion about the cyclicAMP signaling pathways, let us familiarize ourselves with some of the terminologies which are used in this discussion. Some of the common terminologies which we will be using again and again during this lecture is receptor. What we mean by receptor in this class is receptor is basically a membrane protein with extracellular and intracellular domains that interact with hydrophobic hormones or ligands and therefore act as the transducer. So, this is the molecule which is usually a transmembrane protein, and it is the one which actually going to bind to the extra ligand or the hormone through the extracellular domain. The transducer is actually the G protein here. So, once the ligand binds to the receptor, it then interacts with the G protein, and it is the G protein which transmits the signal further, and therefore, this is the transducer for that particular receptor. Now, how does the G protein act as a transducer? It actually goes and interacts with the primary effector molecule, and in this case the primary effector molecule is adenylyl cyclase. Once the ligand interacts with the receptor, the trimeric G protein dissociates, the alpha subunit now comes and binds to adenylyl cyclase, and adenylyl cyclase now in the GTP bound form, G alpha subunit now activates adenylyl cyclase, and therefore it results in synthesis of cyclicAMP, and cyclicAMP therefore is the second messenger. So, the first messenger is the hormone which actually bound to the receptor. Now, what does cyclicAMP do? It now acts on a secondary effector molecule, and in this case it is the protein kinase A. So, cyclicAMP goes and binds to the regulator subunits of protein kinase A, and therefore the catalytic subunit is now separated, that now goes and does a number of physiological processes. Now, cyclicAMP is produced by a number of signaling pathways. Some of them I have discussed here, and what we will discuss today is about the GPCR pathways. In addition to that, you have alpha beta adrenergic receptors, growth factor receptors, corticotropin releasing hormone receptor, glucagon receptor, DCC or dilated colorectal carcinoma. All these receptor pathways ultimately result in the synthesis of cyclicAMP as a second messengers. So, whenever these pathways are activated by their cognate ligands or hormones, it results in the synthesis of cyclicAMP, which then acts as a second messenger. So, neurotransmitters, hormones, inflammatory stimuli, stress, epinephrine, norepinephrine, and a number of other molecules activate the G proteins through their receptors such as the GPCRs, alpha beta adrenergic receptors, and so on and so forth. Whereas the ligands such as glucagon, urocortins, nectin-1 either directly regulate the activity of adenylyl cyclases, or they activate via G protein activation. Now, what are these adenylyl cyclases? There are basically two types of adenylyl cyclases. The previous class we basically discussed about the membrane bound adenylyl cyclases, or this is known as the TM adenylyl cyclases. There are also what are known as the soluble adenylyl cyclases, and in the humans there are about 9 transmembrane adenylyl cyclases or TMACs and 1 soluble adenylyl cyclase or SAC. Now, what do they do? The transmembrane adenylyl cyclases or the membrane bound adenylyl cyclases are the ones which are regulated by the heterotrimeric G proteins, therefore are very important for the current discussion which involves signal transduction pathways leading to gene activation. Therefore, the transmembrane adenylyl cyclases are regulated by heterotrimeric G proteins in response to stimulation of various types of the G protein coupled receptors, and therefore play a very important role in the cellular response to extracellular signals. Whereas the soluble adenylyl cyclases, which primarily occurs in mature spermatozoa, is insensitive to G proteins. So, G protein signaling takes place primarily through the membrane bound adenylyte cyclases. The soluble adenylyte cyclases has nothing to do with the G protein signaling pathway. The soluble adenylyte cyclases are directly activated by calcium ions and bicarbonate ions, therefore rendering the soluble adenylyte cyclase as a very important intracellular metabolic sensor. So, these two perform totally different functions. Together, the transmembrane adenylyte cyclases and soluble adenylyte cyclase regulate a diverse set of essential biological processes which include differentiation, gene transcription, and therefore make cyclic AMP signaling a very important mediator of intra and extracellular signals in eukaryotes as well as in prokaryotes. In prokaryotes, we are already studied in the first class how cyclic AMP through the involved regulates glucose metabolism in cells, prokaryotic cells like E. coli. The main targets of cyclic AMP are protein kinase A, GTP exchange protein. The exchange protein activated by cyclic AMP are EPACs, and the cyclic nucleotide gated ion channels are the CNG. But for today's discussion, we will confine ourselves to how cyclic AMP activates protein kinase A, and then how does protein kinase A activate specific gene expression programs. Now, protein kinase A is the major readout or the primary mediator of cyclic AMP, biological effect of cyclic AMP, and therefore is the predominant cellular effect of cyclic AMP. Now, pKa is not just present diffusely in cytoplasm. pKa is actually tethered to specific cellular locations by a class of proteins known as A kinase anchored proteins. As I said in the beginning, compartmentalization has played a very important roles, and many of these molecules, keeping many of these molecules like protein kinase A in specific locations is a very important part of regulatory cascades, and one group of protein called the A kinase anchored proteins or ACAPs actually keep these pKa bound in a bound form in specific locations inside the cytoplasm. Targeting of pKa isozymes by ACAPs is very important for a number of physiological processes such as cyclic AMP regulation of ion channels in the nervous system, regulation of cell cycle, chromatin condensation and decondensation, nuclear envelope assembly and reassembly, steroidogenesis, reproductive functions, immune responses, and many other intracellular transport mechanisms. So, keeping this pKa in a tethered form by these ACAPs plays a very important role in a number of physiological processes. Now, protein kinase A, this we have discussed in the last class itself, also known as the cyclic AMP dependent protein kinase or the CAPK, transfers inorganic phosphate from adenosine triphosphate to the hydroxyl group of a serine and threonine in a particular amino acid sequence, and usually the sequence which is recognized by protein kinase A is a motif called arginine proline serine or arginine. The instead of proline can be also be other hydrophobic amino acids like glycine, and in this sequence the serine actually gets phosphorylated. So, the consensus sequence for recognition by protein kinase A is arginine proline series. If you look at all the proteins which are targets for protein kinase A, they invariably contain this consensus sequence. So, pKa recognizes this sequence and specifically phosphorylates this particular serine residue. And the protein kinase A in the resting state comprises of two catalytic subunits and two regulatory subunits, and usually they are designated as R2C2. As long as the catalytic subunits are associated with regulatory subunits, it is in an inactive form. Now, what does cyclic AMP do? The regulatory subunit of protein kinase A contains a pseudo substrate sequence like the substrate domain of a target protein, but with alanine substituting serine and threonine. And as you can see, alanine cannot be phosphorylated, and therefore it remains inactive. The pseudo substrate domain of the regulatory subunits which lacks a hydroxyl that can be phosphorylated binds the active site of the catalytic subunit, thereby blocking its activity. So, this is how when catalytic subunit is associated with the regulatory subunit, the protein kinase A remains in an inactive form. Now, once the 4 molecules of cyclic AMP bind to 1 molecules of the inactive pKa consisting of 2 regulatory subunits and 2 catalytic subunits, and as you can see 4 molecules of CAMP can bind to the regulatory subunit, and once it happens a conformational change is induced in the regulatory subunit, and as a result the regulatory subunit can no longer interact with the catalytic subunit, and the catalytic subunit is now free, and it now goes inside the nucleus and recognizes the target sequence arginine proline serine in target proteins and phosphorylates the serine residue. So, in the inactive form of the cyclic AMP protein kinase or the protein kinase A, and once cyclic AMP binds, the regulatory subunit undergoes conformational change, and therefore it no longer interacts with catalytic subunits. Now, the catalytic subunits are free inside and it goes and does its job. So, following activation by cyclic AMP, the catalytic subunit of protein kinase A enters the nucleus and regulates the function of at least 3 transcription factors belonging to what is called as the ATF Krebs family. Now, what are these? CREB, as we already discussed in number of other classes previously, CREB refers to cyclic AMP response element binding protein. There are also many variants of CREB. That is why I have mentioned here that it belongs to transcription belong to the ATF CREB family. There are many members of this CREB family. One of them is CREB, the others are what is called as a CREM, cyclic AMP response element modulator, and activating transcription factor 1 or ATF. Now, what are these? Let us now discuss in detail what are these transcription factors and what is their biological functions. I mentioned some of them here. Here is the CREB. As we had mentioned in the previous class when we discussed in detail about DNA binding domain and transcription activation domains of transcription factors, CREB is a transcription factor which contains a very interesting DNA binding domain consisting of a basic region and a leucine zipper. The leucine zipper is actually responsible for bringing two monomers of cyclic AMP together, so it can bind as a dimer and then facilitates the basic region so that it can now interact with the DNA. So, the leucine zippers are important for bringing two monomers and facilitate the DNA binding domain to interact with the DNA sequence, and many proteins like the CRAB, C-Jun, FOS, all these things contain these leucine zippers and therefore can bind to DNA as dimers. So, the DNA binding domain of the CRAB BATF family of transcription factors contain a basic region and leucine zipper. The transcription activation domain contains a glutamine rich regions called designated as Q1 and Q2. And within this glutamine rich region lies the target for protein kinase A, the serine 133 in the case of CREB, the serine 117 in the case of CREM, and serine 63 in the case of 81 is the target for protein kinase A phosphorylation. So, all these members of the CREB BATF family of transcription factors contain a DNA binding domain consisting of a basic region and leucine zipper, and it contains a transcription activation region which is rich in glutamine residues, and a serine residue within this transcription activation domain is phosphorylated by protein kinase A, leading to the activation of transcription. Now, let us discuss in detail what exactly is the biological functions of CREB and how does it act. Now, CREB specifically binds to an element known as cyclic AMP response element in the promoter regions of its target genes, and this cyclic AMP response element primarily consists of the sequence TGA CG-TCA. And it is very interesting that instead of TGA CG-TCA, if this sequence becomes TGA CTCA, that is, the G is removed, this now becomes the target sequence for a different kind of a transcription factor called AP1, which consists of C-Jun and C-FOS. And we will discuss those things when we talk about protein kinase C signaling pathways. So, you can see one nucleotide difference can change the target specificity of a DNA sequence from CREB to that of another transcription factor. So, the CREB actually binds a cyclic AMP response element which consists of TGA CG-TCA, but if this sequence now becomes TGA CTCA, it becomes a recognition sequence for some other transcription factor. So, all genes which are responsive to cyclic AMP or which need to be activated in response to cyclic AMP must contain this kind of a targeting sequence, which is now recognized by the cyclic AMP response element binding protein. As I told you, because of the presence of the leucine zipper, the CREB actually binds to this target sequence as a homodimer. Now, what are the various events that actually can lead to the activation of CREB? Let us just recapitulate what all will happen. Once a hormone or a ligand, whether it can be epinephrine, whether it can be glucagon or it can be ACTH, once these ligands bind to their cell surface receptors, it results in the activation of the membrane receptor and activation of the G proteins, and this G protein trimer, now the alpha subunit of the trimeric G protein now dissociates and then stimulates the adenylated cyclase, and GTP hydrolysis results in the synthesis of cyclic AMP from ATP by the adenylated cyclase. Now, the cyclic AMP now goes and binds to the regulatory subunits of the protein kinase A, therefore dissociating the regulatory subunit from the catalytic subunits. The catalytic subunit is thus liberated. Now, the protein kinase A catalytic subunit migrates to the nucleus, phosphorylates the serine residues of the arginine proline serine or arginine glycine serine sequence in target proteins like cyclic AMP response element binding protein CREB or CREM or ATF and so on and so forth, which is present in the transcription activation domain. And once CREB is phosphorylated, in the phosphorylated form, CREB is now recognized by a histone acetyltransferase called the CREB binding protein or CBP, and once CREB binds to the CBP, CBP acetylates the histones in the immediate vicinity and makes the histones fall apart, thereby facilitating the recruitment of TAFs and the pre-initiation complex, and therefore the transcriptionation can take place and all the genes having cyclic AMP response element now will get activated. So, this is the basic scheme of transcriptional activation by CREB or cyclic AMP response element binding protein. I have designated the same thing in a schematic form. It's a very, very important, that's why I don't mind repeating this signaling pathway. So once the hormone binds to it, it's a cell surface receptor, which what we are discussing now is the GPCR, which contains the 7 transmembrane domains. And once the hormone binds to a GPCR, corresponding GPCR, it activates the G protein, trimeric G proteins. The beta gamma subunits of G protein dissociate from the alpha subunit. Alpha subunit now goes and activates adenylate cyclase. Adenylate cyclase now synthesizes cyclic AMP. Cyclic AMP now goes and binds to the regulator subunits of protein kinase A, thereby dissociating the catalytic subunit. The catalytic subunit now goes inside the nucleus and phosphorylates the specific serine residues of CREB, and once CREB is phosphorylated, it now interacts with the CBP or the P300, which is a histone acetyltransferase. Now CREB now facilitates recruitment of the pre-initiation complex, therefore transcription activation takes place. So this is a general scheme which involves transcription activation by cyclic AMP. Now the CREB protein has a modular structure with distinct domains, which, as I discussed earlier, it contains the basic leucine zipper motif which mediates dimerization and DNA binding, while the glutamine rich domains Q1 and Q2 and the kinase inducible domain, which is the target for the CBP, constitutes the transcription activation domain of cyclic AMP responsible binding protein. Now these different domains can recruit distinct protein that can modify the transcription activity of CREB. Okay. Now so far we have been saying only CREB, but CREB is not just one protein, but there is a family of proteins which are actually produced by which belong to the ATF CREB family of protein, and many of variants of the CREB, which can be CREM or ATF1 or molecules called as isers, all these things actually arise out of a single gene by alternate splicing. So in this schematic I have actually shown the various proteins which can arise from CREB by alternate splicing, and some of them contain only the DNA binding domain, some of them contain only the transcription activation domain, and so on and so forth, and all of them are actually very important regulators of cyclic AMP response. We will not discuss all of them, but we will briefly discuss some of these variants of CREB, so that we will have some idea how the cyclic AMP response is governed by a family of proteins belonging to the ATF CREB family. I especially want to pay attention to one particular variant of this CREB family, namely the iser, which actually act as a negative regulator of cyclic AMP response. Therefore, we will discuss it in more detail in the next few slides. Now the CREM gene, which is the cyclic AMP response modulator, which is schematically shown in the previous slide, which is slightly different from the cyclic AMP response, the CREB in terms of homology, because certain domains it does not contain certain domains which are present in the CREB. Now CREM actually is transcribed from two different promoters called P1 and P2. Now P1 is a housekeeping promoter that directs the expression of several transcription activators belonging to the CREM family, namely CREM-TAW, CREM-TAW1, CREM-TAW2 and CREM-TAW alpha, as well as the expression of several transcription repressors like CREM alpha, CREM beta and CREM gamma. Now I will not go into details of what each one of them do. There are certain reviews which I have listed at the end of this lecture, and one can go and look up at these reviews and try to understand what exactly is the functions of these different variants of CREM. Now most CREM isoforms emanating from P1 are regulated by pKa phosphorylation, because they all contain the target sequence for pKa. Now the P2 promoter is strongly induced by cyclic AMP by virtue of 4 cyclic AMP response elements which are present in the P2 promoter. So the CREM gene contains two different promoters. When transcription activation takes from P1 promoter, this CREM is constitutively produced, whereas when transcription activation takes from the P2 promoter, it is inducible promoter because it contains cyclic AMP response element, therefore it is a cyclic AMP inducible promoter. The induced isoform, so when the P2 promoter is activated, a different form of CREM is synthesized, which is actually now called as ICER, which actually refers to inducible cyclic AMP early repressor. It actually consists only a DNA binding domain, and therefore functions as a very powerful repressor of cyclic AMP mediated gene expression and is not directly regulated by pKa phosphorylation. If we just go back and look at the schematics here, you can see the ICER contains only the DNA binding domain of DNA binding domain, and therefore it does not contain the transcription activation domain, and therefore it can only bind DNA, but it cannot be phosphorylated by pKa, therefore it cannot interact with the transcription CBP, and therefore it cannot activate transcription. So the induced isoform, namely the inducible cyclic AMP early repressor, essentially consists of a DNA binding domain, therefore functions as a powerful repressor of cyclic AMP mediated gene expression. The very interesting aspect about the ICER mechanism of activation is that the ICER constitutes a negative autoregulatory control mechanism by repressing its own promoter. Because it is a cyclic AMP inducible promoter, so once cyclic AMP produces pKa, and pKa binds to these 4 cyclic AMP response elements in the ICER promoter, and synthesizer, the ICER which is actually made goes and represses its own production. And at least 4 isoforms of ICER, namely ICER 1, ICER 1 gamma, ICER 2 and ICER 2 gamma, collectively referred to as the ICERs, function as negative regulators of cyclic AMP response. So remember, when we say cyclic AMP response and transcription, it consists of not only cyclic AMP response element binding protein and molecules like CREM, it also contains a very important negative regulator of cyclic AMP response, namely ICER, and the ICER is actually produced from P2 promoter of the CREM gene, and this is a cyclic AMP inducible promoter, and since ICER contains only the DNA binding domain and lacks the transcription activation domains, ICER functions as a negative regulator of cyclic AMP response. The ICER comprises only the B-zip domain of the CREM, and its increased expression following cyclic AMP elevation is driven by multiple CR elements within an intronic promoter named a CARE, cyclic AMP regulatory element. Once induced, the ICER localizes to the nucleus where it binds to target DNA, and therefore, since it lacks the transcription activation domain, functions as a very powerful repressor of CRE driven transcription. So, if transcription is activated from the P2 promoter of CREM, it results in the synthesis of a very powerful negative regulator of cyclic AMP response, namely the ICER. However, effects of ICER are transient, since ICER itself can bind to the CARE and repress its own transcription. ICER plays a very important role in the CD4 positive T cells, where it attenuates chemokine and cytokine induction following antigen stimulation, as well as in cardiac myocytes, where blockade of the cyclic AMP response element mediated transcription attenuates the hypertrophic response. So, the ICER plays a very important role in the regulation of antigen stimulation in the lymphocytes, in the T cells, as well as in the cardiomyocytes. A very nice article by Sasson Karsis group actually talk about the inducibility and negative autoregulation of CREM, first published in 1993 in Cell, where it actually demonstrates an alternative promoter directs the expression of ICER, an early response repressor. So, this is the landmark paper that led to the discovery of an ICER and how ICER actually functions as a negative regulator of cyclic AMP response. This is just a cartoon which actually again tells you how important the ICER. We have already discussed this cartoon in the previous slide. The only thing now I am adding here is that when ICER expression is induced instead of CREP, ICER now goes and binds to the cyclic AMP response, and since ICER contains only the DNA binding domain, it cannot be phosphorylated by protein kinase A, and therefore it cannot interact with CBP, and therefore ICER acts as a negative regulator of cyclic AMP response elements. So, when CREP or CREM binds to the cyclic AMP response element, it results in activation of transcription. When ICER binds to the cyclic AMP response element, it results in negative regulation of, or it does not result in the activation of the cyclic AMP response element genes involved in cyclic AMP response. Now, so far I have been telling you that CREP is activated primarily by protein kinase A. So, cyclic AMP is synthesized in cells, cyclic AMP activates protein kinase A phosphorylates CREP, and therefore CREP now activates all the cyclic AMP responsive genes. But in addition to the pKa pathway involving the GPCRs, CREP can also be activated by a number of other pathways. So, here is an example where multiple signaling pathways can lead to the activation of a single transcription factor. We have not discussed in detail the other signaling pathways, so we will discuss these in the later classes, but this is a very important aspect that one has to remember, that when in the biological systems, any transcription factor, more as a rule than an exception, are usually activated by a number of signaling pathways. For example, in addition to the hormones through the GPCRs, annulate cyclase, cyclic AMP and pKa, CREP can also be activated by in the neuronal cells whenever there is a synaptic activity, and this synaptic activity which involves specific neurotransmitters. When neurotransmitters are released near the synapses, these neurotransmitters cause release of calcium ions, and this calcium ions then activate calmodulin, and calmodulin now goes and activates a kinase called calmodulin calcium dependent calmodulin kinase 4, and this calmodulin kinase 4 also can go and phosphorylate the same serine residue which is phosphorylated by protein kinase A, and therefore CREP can be activated not only by cyclic AMP dependent protein kinase A, but also through the calcium calmodulin pathway. So, you can see two different kinases activated by two different signals can activate the same transcription factor. In addition, growth factors can activate CREP through specific kinases like the and specific stress signals can also activate through MAP kinase pathways CREP. We will discuss in detail what these different growth factor signaling pathways and MAP kinase pathways in the next couple of lectures when we discuss other signaling pathways that activate transcription factors. Just remember, this is a very important point. The take home message is CREP can be activated by multiple signaling pathways like the PKA pathway, the calcium calmodulin pathway, the receptor tyrosine kinase or the growth factor signaling pathway, as well as through the MAP kinase pathway, and different kinases can go and phosphorylate either the same serine residue or different serine residues, leading to activation of the CREP function. And normally the level of intracellular cyclic AMP is regulated by the balance between the activity of two types of enzymes, namely adenylate cyclases and the cyclic phosphodiesterases, chiefly in response to hormones and neurotransmitters. So far we have been talking only about the positive regulation, that is activation of adenylate cyclase leading to synthesis of cyclic AMP, activation of protein kinase and so on and so forth. But since cyclic AMP has very potent second messengers, as soon as cyclic AMP is synthesized, these cyclic AMPs are subjected to or cleaved by phosphodiesterases and therefore get immediately inactivated. So, the cyclic AMP signaling is involved in controlling a number of cellular processes, especially exocytotic events involving release of molecules outside the cell in polarized epithelial cells, and this has very important implications in diseases like diabetes, like insulin secretion for example is regulated by cyclic AMP, hypertension, gastric ulcers, thyroid disease, diabetes mellitus and asthma. So, a number of these diseases which involves exocytosis, and these exocytotic events are controlled by cyclic AMP. Heterologous sensitization of cyclic AMP signaling contributes to fundamental physiological processes like timing of circadian rhythms, sexual behavior, neurotransmitter crosstalk, and also a number of neurological disorders such as drug abuse and drug induced dyskinesias and so on and so forth. So, what I am trying to tell you from these things is that cyclic AMP plays a very important regulatory role in a number of physiological processes, which primarily involve not only transcription, but also a number of other cellular processes. It can be release of secretory granules, it can be activation of metabolic enzymes and so on and so forth. But since this lecture series is primarily about regulation of eukaryotic gene expression, we are going to confine ourselves to primarily the mechanisms by which cyclic AMP activates gene expression. So, we are not going to talk about other cellular processes which involve activation of metabolic enzymes or activation of calcium release and so on and so forth. We will focus only about regulation of gene expression. This is the complicated style which actually borrowed from the, which was actually purchased from the SA Biosciences here, which actually tells you how complicated is the cyclic AMP signaling pathway. So far I have told you that a ligand binds to a GPCR, results in the activation of a trimeric G protein, adenylate cyclase is activated, and then CAMP is synthesized, it activates protein kinase phosphorylates CREP family of transcription factors. But if you now see the various physiological effects that cyclic AMP can bring about, you can see how complicated is the cyclic AMP signaling, and this entire pathway we are only discussing one particular aspect in this lecture series. So, there are a lot of things that cyclic AMP does in the signal. So, those who have time can freeze this slide and really go through and try to understand what are the various things that cyclic AMP does. I will not go through the complicated signaling pathway, but I am going to confine myself, as I shown here, just one part of this complicated signaling pathway, namely once a hormone binds to a GPCR, it results in the activation of the G protein, resulting in the synthesis of cyclic AMP by adenylate cyclase. Cyclic AMP now comes and phosphorylates, binds to the inactive protein kinase. Now, protein kinase, catalytic subunit of protein kinase now comes inside the nucleus and phosphorylates a number of transcription factors. So, this particular pathway is just one aspect of what is shown in the others. So, in this entire complex events that cyclic AMP mediates inside cells, we are now going to talk, we are talking only about one particular pathway which I have highlighted in this slide. So, I am emphasizing this time and again, do not think that all the physiological effects of cyclic AMP are primarily due to activation of gene expression. There are also many regulatory events that takes place. Many events that are activated by cyclic AMP involving cyclic AMP involves does not involve activation of gene expression, but involves a number of other cascades, and cyclic AMP pathways also there is a crosstalk between other signaling pathways, as I showed in the previous slide. There is a lot of crosstalk between other signaling pathways. So, in vivo the cyclic AMP response is a very complicated network of events. But since we are going to focus primarily on the gene regulatory mechanisms, we are confining ourselves to one cyclic AMP synthesis in the cells. How does cyclic AMP activate gene expression? So, we are only focusing on one aspect of this entire series of events that take place that can be regulated by cyclic AMP. So, in this lecture we have confined ourselves to the role of cyclic AMP and PK in the regulation of gene expression, and I have skipped a lot of other things that cyclic AMP does. Although serine 133 is phosphorylated by CREB, the key event in cyclic AMP signaling is the phosphorylation of serine 133, so that the CREB can interact with the CBP resulting in activation of gene expression. But turns out only a small fraction of target genes, there is less than 2% of the genes, this event happens, suggesting that in addition to the phosphorylation of the serine 133, interaction of CREB with other regulatory partners play a very important role in the actual activation of transcription of cyclic AMP responsive elements, cyclic AMP responsive genes. In other words, all the events that take place in response to cyclic AMP cannot be explained by the serine 133 phosphorylation alone. Phosphorylation of serine 33 is just one aspect of the cyclic AMP response. There are a number of other things that actually are essential in order for this cyclic AMP response to be manifest in the form of gene activation. For example, CREB activity is regulated by a family of latent cytoplasmic coactivators called as TORCs, which translocate in nucleus, bind to CREB in response to specific extracellular stimuli. The regulatory effects of these and other family members of CREB, such as ATF and CREM on cellular gene expression, are not insignificant. So, remember, although I have been focusing time and again that phosphorylation of the serine 133 by pKa is a primary event in the cyclic AMP signaling pathway leading to gene activation, there are many other events which are associated with this gene activation, which also play a very important role in the gene activation by cyclic AMP responsive genes. In fact, genome wide studies put that the number of target genes which are activated in response to cyclic AMP and CREB binding is approximately about 5,000 genes. So, when cyclic AMP levels go up inside the cells, approximately 5,000 genes can be activated, but this number can vary from cell type to tissue type and so on and so forth. This is just an approximation. So, assuming there are about 30 to 50,000 genes in a mammalian cell, 5,000 is a reasonably high number. So, a significant number of genes are activated by our targets for cyclic AMP response element binding protein. A major point to remember is that CREB family contains not only activators but also repressors. Like I told you, molecules like ICER, CREM alpha, beta and gamma, they actually function as repressors. So, do not assume that whenever there is an increase in cyclic AMP levels inside the cells, it results only in the activation of transcription. It also can result in the repression of transcription, especially if the cyclic AMP increase in cyclic AMP levels leads to the expression of proteins like CREM alpha, beta, gamma or ICER. It can actually result in negative regulation of cyclic AMP response element genes. As I mentioned earlier, CREM gene codes for at least 4 different factors that block CRA dependent transcription, namely the CREM alpha, beta and gamma proteins as well as ICER. So, among the various members of the ATF CREB family of transcription factors, CREM alpha, beta, gamma and ICER actually act as negative regulators of transcription. And if you remember what I told you earlier, the P2 promoter of the CREM actually contains a cyclic AMP response element, and therefore, when cyclic AMP is induced, CREB can actually go and bind to these cyclic AMP response elements, induce the expression of ICER, and therefore, ICER can now compete with CREB, and therefore, when ICER binds, it can actually function as a negative regulator of, there is a competition between CREB and ICER leading to down regulation of the cyclic AMP response. Most of these isoforms are generated by alternate splicing. I have showed in a schematic how these are all generated. Another common feature of many CREB family genes is that these alternate spliced isoforms have different properties, and therefore, their expression can be developmentally regulated. So, here is a very wonderful example of how from one single gene a number of different proteins can be synthesized, and each protein has a different physiological function. So, from a single gene, by alternate splicing, many variants of a transcription factor are synthesized, and some of these variants function as positive regulators, some of these variants function as a negative regulators. So, you can see one gene, one protein is not the rule anymore. By mechanisms like alternate splicing, one gene can give rise to many proteins, and these proteins can have in fact quite opposite functions. Some of them can act as activators of gene expression, some of them can function as negative regulators of gene expression. There are two, in fact, not only alternate splicing, I also told you by utilizing two different promoters, either P1 or P2, you can generate different kinds of variants of the CREM proteins. So far we have been discussing the mechanism by which the CREP transcription factor is activated by cyclic AMP and protein kinase A. In addition, like I also showed you a schematic saying that CREP can be activated by many other mechanisms, and the second pathway that leads to CREP activation is by increase in intracellular calcium. I have shown you in the schematic how, especially in the nervous systems, whenever there is a activation of a synapse by molecules like serotonin, for example, which is a very important neurotransmitter, when these neurotransmitters are released near the synaptic junctions, it can actually activate result in the release of calcium, and as I have discussed here, synaptic in the neurons like the synapses mediated by NMDA receptors or the N-methyl deaspartate receptors as well as the L-type calcium channels, there is an increase in the intracellular calcium, and this intracellular calcium may lead to phosphorylation of CREP via the calcium calmodulin kinase family of serine threonine kinases. This CAMP kinase family is another huge family of protein kinases which play a very important role in the regulation of a number of enzymes as well as genes, and one of them is also a CREP. So, CREP can be phosphorylated not only by cyclic AMP mediated protein kinase A, CREP can also be phosphorylated by calcium mediated CAMP kinases or calmodulin kinases. CAMP kinase 1, CAMP kinase 2 and CAMP kinase 4, all of them are capable of phosphorylating CREP at the same residue as that of protein kinase A in vitro. However, CAMP kinase 2, in addition to phosphorylating CREP at 133, also phosphorylates CREP at serine 142, and phosphorylation of the serine 142 inactivates the transcription activating properties of CREP. So, you can see how beautifully the regulation takes place. When serine 33 is phosphorylated by either CAMP kinase 2 or by protein kinase A, it results in the interaction of CREP with P300 resulting in the activation of transcription. But when CAMP kinase 4 phosphorylates serine 142 of the CREP, it actually results in repression of transcription, because this serine 142 prevents dimerization of the two CREP molecules, and therefore CREP cannot dimerize, and therefore it cannot activate transcription. So, you can see how phosphorylation by two different kinases can regulate the activity of a transcription factor. When pKa and CAMP kinases phosphorylate serine 133 of CREP, it results in the it facilitate interaction with CBP resulting in activation of transcription, but when serine 142 is phosphorylated by a specific CAMP kinase, it can actually prevent dimerization of CREP and therefore results in repression of transcription. So, phosphorylation of a transcription factor, depending upon which residue is getting phosphorylated, can either function as an activator or can function as a repressor. So, this is a schematic that actually shows that how calcium can activate CREP. When calcium is released or when there is an intracellular increase in calcium levels, calcium activates calmodulin, calmodulin now activates the calmodulin kinases or the CAMP kinases. CAMP kinases now go and phosphorylate multiple targets including serine 133 of the CREP. So, in addition to cyclic AMP and protein kinase A, calcium through calmodulin kinases can also activate CREP on cyclic AMP responsive genes. A third pathway by which CREP may be activated involves a cascade of kinases activity initiated by nerve growth factor involving the RAS pathway. In the schematic I had shown in the earlier slides, the third pathway involves the RAS pathway which involves growth factor signaling. We will discuss the growth factor signaling in the later series of these lectures, and one of these downstream substrates of this growth factor receptor pathway is a specific kinase called a 90 kilodalton ribosomal S6 kinase 2 or RSK2, and when nerve growth factor binds to nerve growth factor receptor, it initiates a phosphorylation cascade, and as a result a kinase called RSK2 is activated, and upon activation this RSKs as well as other kinases called extracellular signal regulated protein kinases ERKs or ERKs, they translocate the nucleus where they phosphorylate CREP at serine 133. So, the serine 133, which is the major amino acid involved in transcription activation of CREP, is a target not only for cyclic AMP independent protein kinase A, it is also a target for CAMP kinases, it is also can be targeted by the extracellular signal regulated protein kinase or ERKs, as well as the ribosomal S6 kinase 2. So, you can see CREB can be activated by several different mechanisms, and the serine 133 of CREB can be phosphorylated not only by pKa, but also by CAMP kinase, but also by certain kinases which are activated by specific growth factor signaling pathways. So, just as phosphorylation of serine 133 seems to be critical for the activation of CREB, dephosphorylation of residue is as important, because once CREB is activated, also has to be inactivated. The level of CREB phosphorylation of serine 133 reflects a balance between opposition action of kinases and phosphatases. While protein kinases phosphorylate the serine 33, there are protein phosphatases, especially protein phosphatases 1 and 2, which actually remove the phosphate, thereby inactivating CREB. So, at any given situation, there is an equilibrium between the kinases and phosphatases. As rapidly the serine 133 is phosphorylated by these kinases, it is also rapidly dephosphorylated, and the equilibrium between this kinase and phosphatases defines how long the signal can be active, how long a cyclic AMP response gene can remain active, depends on the balance between the activation of phosphatases and kinases. So, dephosphorylation of CREB at 133 may be initiated by the activation of calcineurin by the calcium carbonyl pathway. So, there are certain pathways which activate kinases, there are certain signaling pathways which activate phosphatases, and therefore, when these pathways are activated, like for example the calcineurin pathway, it actually stimulates the activity of phosphatases and therefore inhibits the CREB activation. The calcineurin may activate nuclear phosphatase PP1 that goes on to dephosphorylate CREB. So, there is a dynamic equilibrium between signaling pathways which activate kinases that phosphorylate CREB, and there are pathways which activate phosphatases which dephosphorylate CREB, and this equilibrium between these two opposing pathways ultimately depends how long the CREB signal, how long the cyclic AMP signal or the positive signal can remain active. Now, we are now going to discuss a very important aspect of cyclic AMP response, especially the role of CREB in the nervous system, especially in long term memory. Now, CREB is thought to be one of the factors necessary for initiating transcription of proteins required for long term memory in a variety of species. So, I am going to spend about 5 minutes trying to explain how CREB plays a very important role in the nervous system, especially in the long term memory. The first study to suggest that CREB is required for memory formation or plasticity was performed in aplysia cultured neurons. It turns out the very elegant experiments were actually done to demonstrate the role of CREB in the long term memory. Long term facilitation was blocked by injection of oligonucleotides containing the cyclic AMP response element sequences into cultured neurons. So, if you now culture neurons from this aplysia, and into the neurons if you simply inject oligonucleotides which contain the cyclic AMP response elements, you can prevent long term memory in this organism. It turns out when you inject this oligonucleotide, what is happening? The CREB now goes on, it is these, like what we discussed about squelching in some of the earlier lectures, these oligonucleotides contain cyclic AMP response elements now sequesters all the CREB protein, therefore, the CREB cannot go and activate genes, is not available for activation of the genes of the cyclic AMP responsive genes. So, by flooding the system with oligonucleotides containing CREB binding sites, you are titrating out the CREB and making less CREB available for activation of the target genes, and as a result there is a lot of loss of long term memory in this organism. Similarly, when you inject a reporter gene driven by a cyclic AMP response element, for example, you take a promoter containing the cyclic AMP response element, hook it onto either luciferase or a GFP or so on and so forth, and if you now inject this cyclic AMP driven reporter gene into these culture neurons, now if you treat these neurons with serotonin, and if you treat pulses of serotonin, and serotonin produces long term facilitation, it also triggers CREB activation. So, whenever the serotonin pulse results in the activation of the long term facilitation, it also you can see reporter gene expression happening. So, you give a pulse of serotonin and you see there is an activation of CREB, clearly indicating that the serotonin somehow results in a chain of events that ultimately results in the activation of the CREB transcription factor. So, a single pulse of serotonin does not produce LTF and does not result in CREB activation, but a repeated pulses of serotonin can result in LTF and also trigger CREB activation. So, experiments such as this described here clearly indicated that neurotransmitters which are actually involved in memory can probably act or functioning through the CREB pathway. So, by injecting oligonucleotides containing CREB binding sites, you can actually lower the memory, and also by you can activate CREB responsive genes or CREB responsive reporter genes by giving pulses of serotonin, clearly indicating that there is a connection between neurotransmitter, CREB activation and memory. Very elegant experiments have been done to demonstrate the relationship between CREB and memory. I have just listed the summary of all these things here. For example, whenever there is an action potential and results in the release of certain specific neurotransmitters like serotonin at these junctions, these neurotransmitters bind to the neurotransmitter receptors, and when these neurotransmitters bind to the neurotransmitter receptors in the synapses, it results in the release of calcium and cyclicAMP, and these activate protein kinases, like for example, cyclicAMP activates adenylate cyclase or calcium activates calmodeling kinases, and they in turn goes and phosphorylate the serine 133 of the CREB, resulting activation of genes whose proteins are actually involved in synapse formation and therefore strengthen the synaptic transmission leading to long term memory. So, here is an example where signaling molecules, neurotransmitters can actually bring about a physical or physiological response, namely long term memory, through the activation of a specific transcription factor. So, it is a very elegant experiments have been demonstrate CREB plays a very important role in long term memory. There are a number of interesting papers which actually discuss how important is CREB in various aspects of nervous systems. Here is a paper which actually demonstrates expression and function of CREB is increased by different types of antidepressant treatment, suggesting that CREB is a common post receptor target for a number of antidepressants. So, there is a lot of interest among neurobiologists to understand the role of CREB in a number of neurological processes like long term memory, in this case for example depression and even drug addiction and so on and so forth. Here is another very interesting paper which says a protein called brain derived neurotrophic factor is a member of the nerve growth factor family, and this growth factor actually targets CREB, okay, saying that the neurotrophic factor synaptic plasticity involves actually CREB. Similarly, upregulation of the cyclicAMP pathway has actually been shown to administer result in the chronic administration of certain drugs which actually involved in certain addiction and so on. So, drugs which actually drug addiction, for example, when you get addicted to drugs, this drug addiction seems to be primarily involved in the activation of cyclicAMP pathway and CREB. And this paper actually very interestingly describes how drug addiction actually involves activation of CREB and increasing intracellular levels of cyclicAMP. So, the cyclicAMP response or the calcium increase coupled to the CREB activation seems to be involved in the regulation of a number of neurological processes inside the brain. So, what I am going to discuss in the next few slides, I have just highlighted some of the important aspects of cyclicAMP regulation and how cyclicAMP is a very important second messenger, and by activating a family of transcription factor belong to the ATF CREB superfamily, regulates a number of physiological processes both in the nervous system as well as outside. So, some of the important points that you have to remember whenever you talk about cyclicAMP response and gene expression, I have just listed in the next few slides. For example, the summary of what I have told you is that whenever a hormone binds to a hormone receptor, this results in the activation of the trimeric G protein, and this results in the activation of an adenylate cyclase. Now, adenylate cyclase now converts an inactive protein kinase into an active protein kinase A. Now, active protein kinase A goes inside the nucleus, phosphorylates the serine 133 of CREB, and this results in the activation of target genes, leading to through the binding of or the activation of the histone acetyltransferase the CBP or P300, resulting in acetylation of histones and activation of transcription. So, this is the summary by which molecules which activate a adenylate cyclase pathway or the PKA pathway result in the activation of the CREB. Now, cyclicAMP, the important things that you have to remember about cyclicAMP is that cyclicAMP is generated from ATP by adenylate cyclase. It is a very popular second messenger involved in the G protein coupled receptor pathway, and cyclicAMP does not function in signaling pathways initiated by receptor tyrosine kinases. It is a very, very important point. We are going to discuss about the receptor tyrosine kinase in the future classes. As remember, cyclicAMP is not a second messenger when the receptor tyrosine kinase pathway is activated, and cyclicAMP is primarily degraded by phosphodiesterases. And I have also mentioned here important point that you have to remember adenylate cyclases. In mammalian cells, there are two types of adenylate cyclases, soluble adenylate cyclase and membrane associated adenylate cyclase, and the membrane associated or TM adenylate cyclases are the ones which are involved in the growth factors, the GPCR signaling, whereas the soluble adenylate cyclases are not involved in the signal transduction through G proteins. They are directly activated by molecules such as calcium ions and bicarbonate ions. Now, what are the targets of cyclicAMP? CyclicAMP is targets the cyclicAMP dependent protein kinases as well as cyclicAMP cyclic nucleotidated ion channels. We have not discussed this aspect because it is not linked to the gene expression. Now, what is the specificity of CAMP signaling? cyclicAMP and pKa are localized in specific regions, and cyclicAMP kinase associated proteins are the A caps anchor inactive pKas to specific subcellular locations, which has a very important regulatory role. Now, what are the cellular functions of pKa? pKa can go and phosphorylate a number of cellular enzymes and regulate their activity. We have not discussed this particular aspect in our lecture series because it is not directly linked to gene expression. Like for example, pKa can activate lipases, glycogen phosphorylases, cholesterol esterase, and so on and so forth. It can inhibit glycogen synthase. It has very important regulatory roles in carbohydrate metabolism and diseases like diabetes and so on and so forth. But what we primarily discuss in this class about the activation of transcription factors such as CREB, CREM, ICER and ATF1, because that is what is relevant to this lecture series. Now, CREB is the primary link between cyclicAMP and gene transcription. We discussed various aspects of the CREB activation. I have listed some of the important aspects like what is the cyclicAMP response element, what is the residue that is phosphorylated by pKa, how does CREB interact with CBP P300, and how does the CBP P300 by virtue of histone acetyl transferase activity acylates histones leading to stimulation of the pre-initiation complex formation and activation of transcription. Now, in addition to the CREB ATF family of transcription factors, pKa also phosphorylates a number of other transcription factors like NF-kappa B, nuclear receptors, high mobility group proteins, and the GLI CRUPL family members, etc. But we have not discussed some of these aspects, and if you are interested, you can go and read up some of these things later by using specific papers. Now, intracellular levels of cyclicAMP is regulated by equilibrium between two activities of types enzymes. Adenylate cyclase act as positive regulators of cyclicAMP response. Cyclic nucleic phosphodiesterases act as negative regulators. So, as soon as cyclicAMP is formed, it is also cleaved by these phosphodiesterases. Now, what are the physiological processes which are governed by cyclicAMP signaling? A number of events are regulated by cyclicAMP. I have listed all these things, but we have discussed only one aspect of this, namely regulation of gene expression. But in addition to that, a number of other processes like the exocytic events, which play a very important number of disease processes, are regulated by cyclicAMP. Now, just to give you that there is also a very important link between cyclicAMP and cholera. Cholera toxin, for example, hexameric protein produced by Vibrio cholerae, it causes cholera. And cholera is primarily caused because the cholera toxin goes and activates a GPCR protein, and it increases cyclicAMP levels in epithelial cells by causing ADP ribosylation of the G alpha subunit, and therefore there is a persistent activation of adenylate cyclase, there is continuous production of cyclicAMP, and as a result there is a massive flow of water from blood intestines leading to cholera. So, cyclicAMP is actually involved in diseases like cholera. So, I suggest that you read up at least 2 of these review articles. There are a number of review articles describing cyclicAMP activation, CREB and so on and so forth, but if you are interested all these things, one can go ahead and read at least 2 of these papers. Mark Montminy is the one who had discovered CREB and has done a very interesting work in the regulation of CREB, and is a very nice article in Annual Review of Biochemistry. And there is also a very recent review article in Cellular Signaling about regulating gene transcription in response to cyclicAMP elevation, and these 2 papers give you much more inputs about the mechanism by which cyclicAMP regulates gene expression. I think I will stop here.